What the tea leaves say, the Roundtable Research. An open tea podcast, communication and dissemination services.
Buongiorno a tutti, siamo presso l'Università Cattolica del Sacro Cuore di Piacenza. Il contesto è un evento ideato all'interno del progetto di ricerca SIRAM, che si occupa di innovazione per l'agricoltura rigenerativa. Apre i lavori il professor Luigi Lucini, docente di chimica agraria, che ci parlerà dell'uso dei biostimolanti per un'agricoltura sostenibile, dalle definizioni alla pratica.
Good afternoon, everyone. I would like to start from the concept of biostimulant. The term is new, and often different terms are used, like biofertilizers. Sometimes you can hear about biostimulants, biosolutions. So they use different ways. We will use biostimulant. That is what, at least in Europe and US, is the official definition now, and there is a general agreement on this term. But the concept is very, very old. The idea of thinking about some complements that are not pesticides and are not fertilizers, but that can still promote, increase the performance in field, starts when we realized that legumes have a symbiosis with microbes. So it's a very, very common story. And this was just the beginning. Then in more recent years, there were some tentative evolutions. And at least like in Italy, the definition was products with specific activity on plants, that is meaningless. And nowadays, we are moving from local national regulations to a common EU regulation that has defined and regulated in EU, and similar is happening in the United States, and regulate this class of compounds. When we say product with specific activity, I was saying is meaningless because they just specify that it's something different from fertilizers, and so are not fertilizers, are not pesticides, but it's known that they improve the fitness of plants. So it was a pretty empty concept, and in Italy at that time, until 10-15 years ago, they were included in the regulation of fertilizers. So they were just a very, very small market compared to the market of fertilizer, market of pesticides, and there was no a specific need in terms of regulating this market. However, scientific literature started to produce a lot of evidence about the advantages of using these type of products, using the most studied at that time were mycorrhiza, that are now well consolidated, or humic acids, what we call generally organic matter, is a fraction of the organic matter. And then started to appear protein hydrolysates, like from animal or from plants, or seaweed extract, the last one. So the scientific evidence was growing fast, and at the same time, the market was growing really, really fast. So together with these objective evidences that this type of products can provide advantages, farmers started to adopt these solutions more and more and more, and the market is growing. Compared to the standard in agriculture, this increase was really surprising. So at the beginning, it was a very small market, and therefore no specific need to regulate this. It was just like a small part. We can include them in biostimulants. This was the approach in the different countries. But then, when this market became, you can see the figure here, really important in terms of volume, in terms of money, and in terms of hectares where these products are used, then the question became not a local question. It's not a matter of Italy. Actually, Southern Europe is using more as compared to Northern Europe. But nowadays, they are really, really spread worldwide. So in both North and South America, in Europe, in Asia, they are growing. And you see, in 2026, the projection is that it can account for $5.6 billion. That if you check in 2018 to 2026, you can easily realize how fast they are growing. So this has been supported by scientific evidences, by the positive feedback from the farmers, and by regulations, because we, I don't want to could take a long story speaking about sustainability in agriculture, but in general, you know that the EU Green Deal and the regulations in general are forcing us to reduce inputs in agriculture. But at the same time, we know that climate change and population growth are demanding an increase of yield. So are two parts that do not match one with the other. And the older paradigm, according to which you increase fertilizers, you increase the yield, and so this is would be the simplest way to think about, but this is not any more sustainable, this is not any more feasible, because the potential of fertilizers to increase yields are getting the top, so we cannot increase. When you double fertilizer, you simply double the yields. It doesn't work this way, of course, and at the same time, increasing fertilizers decreases nutrient use efficiency, decreases the efficacy of these products. That means the environment can be negatively affected because the excess of nitrogen can reach the drinking water. For instance, you know that in Europe we have a limitation for nitrate, for the amount of nitrate in drinking water. So you cannot simply think to increase the inputs, but it could be a smarter way to think about strategies that increase the efficiency of the inputs instead of increasing the inputs. And this has been driven, but those are the requirements of the Green Deal in Europe. So the target was to reduce, staying in fertilizer, not to increase, but even to reduce by 50%. That was totally non-realistic when they set this limit. Regardless the limit that has been political and non-technical choice, still the mandate is to reduce inputs, to increase sustainability, to increase long-term health in soils. And biostimulants can work this way. The first attempt that started to define biostimulants as they are now has been provided by an association of producers. When the market has grown a lot, at the same time, the fair producers, they started to make a lobby and asking the European community, please regulate this market, because now the market is big enough. There are so many players. Maybe some player play in a very rigid and rigorous, scientifically supported way. Maybe some other are just trying to get some money in the short term and then they disappear from the market. And this was posing problems in the market, because when you think about the perspective of the farmer, for the farmer it's very difficult to discriminate. This product that is really working, has been tested in different conditions, so can enter the market from another one that a different producer, maybe a very small local producer, just sell in short term, and he knows that it will stay on the market for 2 seasons, 3 seasons, whatever it is, and then he leaves. So the pressure to regulate the market was from the producers, and they established the EBIC, that stands for European Biostimulant Industry Council, so the association of producers and stakeholders that are interested in this area, and they started defining what biostimulants are. This is the pretty good definition. I start from the bottom, because the bottom helps us to clarify. Biostimulants do not have significant nutritional effects, are not fertilizer, therefore. You're not providing nitrogen or phosphorus, whatever. If you apply, like for instance, protein hydrolysase, they are rich in amino acids, so they are a good source of nitrogen. But if you apply 1 kilogram per hectare, means that the nitrogen you provide is meaningless in terms of nutrition. So this is an example where they are not fertilizers. And second one, they have not to be pesticides, so are not used for pests. And these definitions are very important because I don't know if you probably know, at least one of the two courses know the process to the regulatory process for pesticides is extremely costly, long, so it's a huge investment, millions and millions and millions. And you understand that if some companies can find a shortcut to enter the market as pesticides, naming them biostimulants would be a mess, because you completely destroy the regulations. So they have not to be pesticides, not active against pests. They have not to be directly providing nutrients. They are the third way, as my colleague Ettore was saying, are a third way, something different from these two. And what they should do, it was not possible to define them according to the classes. We will screen some of these biostimulants. There are so many types. So the definition was on the activity rather than on the composition. And the activities that they should, when applied, so first, natural products or microbes, so two classes. And what they should do is, when applied to plants or to rhizosphere, they should stimulate the natural processes of plants to increase the uptake and the efficiency of use of nutrients, to increase the tolerance to abiotic stresses, or to increase the quality of the product. This was a definition that came at that time. I was in the commission that was defining these rules, and has been taken by literature. So we sat down and say, okay, which are the advantages that they normally provide? Are those reported here that were also used as a basis for the EU regulation that I'm going to speak about in a very few minutes. At the same time, in the United States, the Biostimulant Coalition, that is the counterpart in Northern America to ABIC in Europe, the Biostimulant Coalition tried to define biostimulants as substances or compounds different from nutrients, so this is in common with Europe, that have been proven to be beneficial to plants. So they provide some benefits. This one, I think, is rather too much generic to regulate a market, but still we are going in, as you can see also here, in a position that is different from fertilizers, so do not directly provide nutrients. Starting from these inputs, the EU recently, because this regulation entered into force since is 2 years, that is in force in Europe, the regulation 1009 from 2019 is a regulation that regulates fertilizers and biostimulants, two different classes, defining what they are, how they can enter the market, and which are the regulations, the ways to prove that this product is really effective. And this regulation confirmed the approach of ABIC because they mainly focused on the activity. A biostimulant, according to the law in Europe, has to be a natural product or a microbe that is able to either increase the tolerance to abiotic stresses, and this is very relevant nowadays with climate change, of course, so abiotic and not biotic, of course, can increase nutrient use efficiency, quality traits, quality means whatever has a value in the market, so can be fruit size, can be firmness, can be color, can be shelf life stability. These are quality aspects for which the farmer receive a money, basically, a concept. Or the last one, that is this fourth was a bit a debate between the people involved, improve the availability of nutrients that are confined in the rhizosphere. Two simple examples, phosphorus and iron. They might be in soil, but might not be available to plants. So you know that, for instance, with phosphorus, you have to check not total phosphorus, but available phosphorus. And very often, these two are completely different parameters. Some, like mycorrhiza, are able to solubilize phosphorus, making phosphorus available to plants. So it's not the amount of phosphate or whatever you apply, but it's rather to increase the ability of the system to efficiently manage the resources that are there. The same for iron, there are two strategies for absorption and so on. This fourth a bit overlaps with nutrient use efficiency. Is not so straightforward to decide, is this process increasing the efficiency of use or the availability? The two come together. But by the way, in both cases, the important concept is that biostimulants do not provide nutrients. They improve the ability to use what is being applied. Possibly what is being applied to a lower amount compared to what we used to do in the past. And this regulation then defined what are product functional categories, the ones here in black. One of the product functional categories are biostimulants, microbial and non-microbial. And for microbial, non-microbial, identified, sorry, this was in Italian because it was a picture, defined the materials according to which these can be used as biostimulants. So are like, just to give you some example, compost, because it's the same in English, but digestate or byproducts of the food industry, microorganisms, polymers, byproducts of animal origin are, or normally industries use byproducts because they are more cost effective. So the products that do not have a high added value, otherwise they enter the market in a different way. And these byproducts are converted into biostimulants. So according to the regulation, you have to prove one of the four that are called claims. So you have to prepare a dossier in which you target one of these claims. So you have to write in the label, this product works to increase tolerance to drought, for instance. A specific claim that has been proved by field trials, has been demonstrated and I will describe later on how. And this product has to be prepared using some of these materials, using natural products. Microbes can be single microorganisms or consortia, and to date, this is a long debate at EU level nowadays, because in the regulation they just added 4 species for microbes, but actually in the market there are much more candidates other than Azotobacter, that is a nitrogen-fixing bacteria, or Mycorrhiza, or Rhizobium, or Azospirillum. These are known since long time, but there are so many others that industries are developing. Now the debate is about how to integrate this list. This list has been talked as a starting point, like for pesticides. There is an annex of positive inclusion of active ingredients. Then you can add to this list new molecules, provided that you have a dossier that supports the inclusion, of course, and the evaluation process supports the inclusion. This was the idea here, but this has not been developed further. So nowadays, for microbial biostimulants, these are the only microbes that are permitted. Of course, possibly, probably, in the next years, this process will be improved, but now has been stopped because the EU has not regulated further how to integrate this list. And, well, this was the part, I was the representative for Italy, because when the regulation has been released, in a regulation are general concepts. So they say you have to prove this claim, but then how to prove it? How to provide an objective evidence that this claim has been achieved? Starting from the regulation, there's ISO standard regulations have been released to explain how to implement the concepts of the regulation, the EU regulation. This is, for instance, the one I contributed to, how to determine quality traits when plant biostimulants are used. So a representative for each country met in Europe and defined in detail how to carry out trials, which are the parameters you can measure, which is the statistical difference that has to be achieved to say, okay, it's different. So any information that can be used to let this applicant prepare a dossier that will be then evaluated by EU and therefore will provide an what is called the CE mark. CE in Europe is, sorry, here no space. When you see for non-Italian, the CE means that the European community has approved it. And for biostimulants, some of them have the CE mark on the label. This means they can be sold whenever in Europe. They pass it through this process. However, there are still on the market, this is the queue of the previous local regulations. On the market, you can find also biostimulants without the CE that have been registered, have been approved according to the national regulations. For Italy, the Decree 75, that was the regulation for fertilizers. But those are just authorizations that are expiring in the next 5 years, whatever it is, the next few years. This regulation, the 1009, will replace national regulations and will be the only way to enter the market. So those are the claims, but how do they work? And why are they interesting? To explain why biostimulants are so interesting, I would like to come back to a bit of plant biochemistry. You know that plants are sessile organisms, cannot escape to stresses. They can only react to limited water, high temperature, high radiation, whatever. They can only react with compounds, with biochemical processes. And for that reason, the plant biochemistry is really complex. What we call secondary metabolism, that is not the primary metabolism, is like photosynthesis, glycolysis, Krebs, the classical biochemistry that is used for plants to grow. But together with that, we have a second part of the metabolism, that is the secondary metabolism or specialized metabolism, somebody call it, that explains how an organism, and in plants this is extremely important, can respond to the environment. And plants have a very broad set of biochemical processes because they have a very well developed hormonal system to respond to like biotic and abiotic stresses. They have mobile RNAs. They have some metabolites that are accumulated, like proline when you are in drought, or polyols like mannitol again in drought. And the signals that are really hormones regulate the metabolism, so regulate how the plant biochemistry works in response to high or low light intensity, in responsible to mechanical stresses, like an insect that is feeding. And here I want, because for instance, jasmonate is normally, in our perception, is normally related, it's just an example, but is normally related to biotic stress. So if you studied plant pathology, you know that jasmonate is one of the first that activate response to pathogen infection. And that's true. What I would like to highlight that is when we think about a regulatory perspective, it's important to say this product works for this abiotic process. So this biostimulant works for chilling injury, protect plants from chilling injury, that there are some on the market. So you're speaking about a very specific abiotic process. However, in plant biochemistry, several of these messengers, these exogenous signals, are in common between abiotic and biotic processes. So, like when you increase phenolic compounds that are very good antioxidants, or steel beans in grape. For the students of viticulture, you know that steel beans are very important in grape and are also linked to human health, but also are phytoalexins, so are involved in plant defense. Resveratrol and other phytoalexins are good antioxidants and also phytoalexins, so are involved both in abiotic stresses, can be drought, and in biotic infection. So this overlapping means that from a biochemical perspective, we cannot simply say, here we have abiotic and here we have biotic. And so many processes are in common. If you think about the infection in plants, it starts with hydrogen peroxide to trigger lignification and isolate the pathogen spread. But hydrogen peroxide, you know, is an oxidative stress. And when plants produce hydrogen peroxide, also produce antioxidants. But antioxidants are also important in drought. So I could provide you so many examples where it's really difficult to separate what is a response to an abiotic process and what is a response to a biotic process. However, so this is biochemistry, but then from a practical point of view, when there is a regulation, you have to use the regulations to enter the market, of course. So what is the difference? The difference is that you cannot claim with biostimulants that this biostimulant works for a pathogen and has to be clear in the label. Then, to some extent, biostimulants also promote defenses. So they have a desired side effect to increase also response to biotic stress. But the main aim has to be abiotic stress, because this is the way you enter the market. So it would be unfair to sell for this one and then ask the farmers. Somebody did, like an example could be laminarin. Laminarin is a polysaccharide that triggers defenses. It's also biostimulants, but it's also good in eliciting defenses against fungal diseases. In grape is registered. Was a biostimulant. When you realized that people were using this one for plant defense, the producer had to prepare the dossier for plant protection products. So changed the market. The same trichoderma. Trichoderma is a very good fungal biostimulant, but it's also a very good control agent. At the beginning was entering the market as biostimulant, but if the main use is for a plant protection product, then you have to comply with the other regulation that is stronger, is more costly, okay, but if those are the regulations, you have to play according to the rules. But, so basically, what is the concept? That you in the dossier for authorization, the trials have to be designed in order to meet the claim you want to write in your label. If the claim is nutritional quality, you cannot check the resistance to powdery mildew. You have to check if the quality can be firmness, can be color, can be bricks, whatever, acidity in case of grape, whatever you are considering, that this specific parameter has been improved by the biostimulant, and stop. This is how it works now. When we look at the perspective of what is called the plant holobiont, you know the term holobiont? When sometimes the term plant holobiont is used to identify plant and the microbe associated more to roots in rhizosphere, but also to phyllosphere to some extent. This complex system is a system where the roots exudate compounds, so actively invest photosynthate, releasing molecules in the small surface of soil that surrounds the roots, and this is done to recruit beneficial microbes. These beneficial microbes provide advantages to plants because, for instance, they can solubilize phosphorus or iron, so they can make iron more available. Or, for instance, they can fix nitrogen, so increase nitrogen use efficiency. Or they can elicit some transporters, high affinity transporters for nitrogen. At the same time, these microbes, when they are in contact with the root, they elicit the defenses. Defenses also against abiotic stresses. It's the same where if you think about the probiotics you take for the health of your intestine, you probably hear so many times that it promotes your immune system. This is exactly the same concept in a completely different environment, but with the differences we have in the two systems, but the philosophy is the same. These microbes at the beginning are perceived as microbes, as they are actually, so potential infection, and therefore plants activate defenses. And those defenses include, you see here, systems that reduce, for instance, reactive oxygen species, so that reduce oxidative stress. And this is the problem when you have drought and salinity. But it's also the problem we have when we have a fungal infection. So, these biostimulants work as elicitors. Elicitors means molecules or microorganisms that can trigger a response, and the response comes, so are perceived by specific receptors. We say that they have hormone-like activity, are not directly the hormone, are not auxins directly applied. Even if synthetic auxins are available and very cheap, but that's not the case, they are not biostimulants. What is a biostimulant is perceived by plant receptors, and in response, plants change their biochemistry. So they work, from a biochemical point of view, they work in a different way. And this new way they switch to is a way that promotes a different expression on genes, and therefore the accumulation of beneficial molecules. That means this plant has been elicited, has been triggered to increase some molecules that are helpful for her to respond to limiting conditions, low nitrogen conditions, high temperature, whatever. And these are secondary metabolites, the ones you can see here. The secondary metabolites, as I was mentioning, they play for biotic and abiotic stress at the same time. So they also increase plant growth. And this is something that normally the farmer likes to see. When he sees his plants are greener and bigger, he feels they are better. But actually, when we use biostimulants, we want to have a better yield, not a better plant. They often go together. So this part is what the farmers are looking at. But this one is what we are actually targeting with biostimulants. When a farmer can see at phenotype level visually that the plant is healthier, then he expects the yield is better. And this is normally the case. Some examples about these biostimulants. So this is the, let me say, the regulatory and philosophy that says, underlies the concept of biostimulants. But you may wonder, okay, but in practical terms, what are they and how can we use them? I'm providing here some examples of some classes of biostimulants because I've been working more on. Humic substances, seaweeds, protein hydrolysis, and mycorrhiza. The first three are non-microbial, the fourth is a microbial biostimulant. And starting from humic substances, I will show you some visual effects that explain why plants can respond in a completely different way. As first, what are humic substances? This comes probably from solid chemistry in your bachelor, but humic substances are an important component of soil organic matter and is basically polymerized form that comes from the degradation of decomposition of cells and using the activity of soil enzymes and soil microorganisms. And that corresponds to three fractions: humins, that is what is non-soluble in water regardless of the pH, humic and fulvic acids. Humic acids are soluble only at low pH level under acid conditions, while fulvic acids are soluble at all pH. These are the three macro classes, and this is a picture that is not representative, it's just to have an idea about the polymerization. The structure is complex, and you might know that these are also part of the soil structure. When this organic matter has to be used as biostimulant, normally cheap and available sources are used. Leonardite, peat, compost, and vermicompost are probably the most common sources of this high molecular weight fraction of the organic matter in soil. So from there, humic or fulvic acids are extracted, are formulated, and then used as biostimulants. This is, in very few words, how the process is. Why are they interesting? again, this is not biostimulants concept, this is probably a bit more soil chemistry, but you know that organic matter and humic acids in particular can improve the cation exchange capacity, and this is due to their complexing action. I kept here in the structure, but if you check, they have like carboxy groups here, so they have negatively charged groups that can have the counter ion, that is a cationic species, so as a consequence, they improve the cation exchange capacity thanks to the carboxy, to the negatively charged groups. At the same time, they can have a buffering effect. And this buffering effect in some soils, especially in alkaline or acid soils, this buffering effect can modulate the availability of nutrients. You know that there are curves according to which nutrients are more or less available, and each of them has a pH range in which it's available. Buffering pH increases the availability of nutrients. That is one of the concepts of biostimulants. And then, since they can complex metals, they reduce, for instance, the toxic effect of sodium cationic species. So under salinity conditions, they are known to improve the response to the stress. So from one side, this is the concept when humic acids are added. The soil particles, soil aggregates have a better structure, and thus better aggregates have an improved ability to exchange cationic species. So soil retains nutrients. Nutrient use efficiency also relates to the ability to retain nutrients, not to be leached away by irrigation water or by rain or by whatever. And the ability to retain is also linked to the presence of humic acids. But besides that, and this is the concept of biostimulant, humic acids are very interesting because some fractions of humic acids. It's not organic matter to improve, are not amended. So it's not soil amendment. We are speaking about low amount with certain specific bioactivity. And this example explains well what I'm meaning. Because if you check the roots there are examples in monocot, in maize, C4, and in tomato, in Arabidopsis, is the reference, in sugarcane. So across pretty different crop species, Arabidopsis is not a crop, crops and Arabidopsis, the effect was consistent with differences from species to species, but you see, when we have humic substances, how root development is different. The root architecture is affected by humic acids, and the reason is that they have the hormone-like activity I was mentioning before. So they are not simply ameliorating soil structure. Yes, they do at high amount, and in that case we call them soil amendment products. In this case, relatively low amount of the soluble fraction. They are often applied, for instance, in drip irrigation, so locally where the roots is developing. And in low amount, they can play this hormone-like activity that changes the biochemistry, changes the phytohormones profile in roots. And when you change the hormones, the growth is different. As a consequence, you see, when we have humic substances, roots are more developed, lateral roots are higher, and root hairs are higher. That means a higher absorption capacity that increases, sorry, I finish just with this concept 1 second, that increases the ability to absorb water, to explore a larger portion of soil, increases the ability to absorb nutrients, and as a consequence, increases what we call it nutrient use efficiency. It is one of the claims, and increases water use efficiency, that is one of the tolerances to abiotic stress. So 2 of the 4 claims we started from can be met by humic substances. So this is a good example about what I was mentioning before. So we are not providing nutrients, we are providing something that changes the metabolism, the biochemistry of plants. And all these cases you see, you don't need we also carried out statistical analysis, of course, but it's very visual. The difference is so clear that simply by comparing the two phenotypes, you can see where humic substances are present and where they are not. And at microscopic level, this work has been done by a colleague of mine in Padua, now she retired. At the same time, if you look at the roots by microscope, you can see how root hairs develop in a completely different way. And you know that root hairs are the active part of roots, are the part that is biochemically active, that is involved in nutrient absorption, that is involved in water absorption, that is involved in recruiting microbes, creating the favorable environment, the holobiont I mentioned before. But an interesting point that has been realized later is that it's not only morphological, the difference when humic substances are used, but the expression of transporters for nutrients. These are nitrate transporters. Some nitrate transporters are constitutive, some other are induced. So some are expressed regardless the level of nitrogen. Some other are expressed when nitrogen is limiting as an attempt to increase the uptake of nitrogen, because plants need nitrogen. Some fraction of humic acids are able to increase the expression of these proteins, these transporters for nutrients. Specific transporters in the root hair surface are expressed in response to the application of humic substances. Again, this means a higher nutrient use efficiency. And there are also good examples in literature about the ability of humic acids to modulate environmental stress. So this is something we cannot see visually. It's something, it's not like root development, of course, that is more intuitive. But in general, when we have stresses, I mentioned it before, hydrogen peroxide is a good example. Both biotic and abiotic stresses act through a mechanism that involves the production of reactive oxygen species that are toxic species. The very most are radicalic species, so are very oxidizing. So plants need to manage these molecules, and normally managing these oxidant molecules means that the plant's biochemistry, the plant performance goes down. It's like when you don't feel well, you cannot perform. The same is for plants. When we have this oxidative imbalance, so when we have a condition where these reactive oxygen species are produced, when we have stresses, and this is the case, then plants' performance decreases. And normally this is a balance, so plants have a way, so these are physiological molecules, are always produced. The problem is when the production of reactive oxygen species is not counterbalanced by antioxidants. In that case, plants perform not so well. Humic acids, together, I have to say, like several other biostimulants, are able to, when they trigger secondary metabolism, they also induce the accumulation of antioxidants and antioxidant enzymes, like peroxidase and catalase, that are used to stabilize, to manage reactive oxygen species, like peroxides and hydrogen peroxide, that are not yet radicals, but they are producing radicals, are pro-radicalic molecules. So the ability to manage these molecules reduces free radicals and in turn reduces plant stress. The second class of biostimulants are seaweeds. This is a large word. I mean, as you can see here, so many different seaweeds can be used. Brown, algae. Now, laminaria, that produces laminarin, the molecule I mentioned before, now is not anymore, is being removed from here because it will be included in pesticides actually. But ascophyllum-based products are very, very, very common on the market, also Clonia maxima. So brown and red seaweeds and also green seaweeds are used Seaweeds are plants, of course, and they are normally harvested. Most of those companies, the biggest one is Italian, has been recently acquired by Syngenta, one of the biggest players on the market that wanted to enter the market of biostimulants, so they bought the leader for seaweeds in Europe. That is a company that was here in central Italy, in Vasto. And so they to enter, like Pfizer and BioNTech, they wanted the vaccine, they bought the company. And his was the same. You want to enter the market with some products that are already on the market. This company had a part in Norway where they harvest the seaweeds and they process here in Italy and they formulate in a proper way. It's a smart company because it produces nice products. And then they had the largest share of the market in Italy and in Southern Europe in general. So they are harvested. Some other companies, like Acadian, is based in Canada and harvest seaweeds there, and then they process in Europe and sell in Europe. So there are several examples, but by the way, in all the cases, seaweeds are harvested directly. So we have different seaweeds. Depending on how you prepare, you have different products. Like for a soil plant, also aquatic plants, seaweeds, they have different profiles that can change with seasonality, can change with plant stage. So depending on which is your source, how you prepare, you extract your components. Like if you extract under alkaline conditions, you can maximize natural auxins, so plant growth promoting hormones. If you extract under higher temperatures, you can extract polysaccharides. If you extract with carbon dioxide, with supercritical carbon dioxide, you can extract small molecules. So you can take different fractions that make different products. So we cannot simply say seaweed contain this class of molecules that work for that. We have to say that there are several different seaweeds, and depending on how they are prepared, then we have different products that have different activities, because of course the profile, phytochemical profile correlates with the bioactivity you can measure. And those are the parameters that determine mainly the profile in seaweeds. The type, the species you are using, when you harvest it in the season, how you extract and how you formulate. One interesting point of seaweeds is that the polysaccharide fraction, that is one of the most relevant, most characteristic in seaweeds, the polysaccharide fractions is able to interact with several signaling receptors in plant, and therefore those polysaccharides activate our response. The example of laminarin before is a good example in this case, because the polysaccharide itself is not bioactive against pathogen or against stresses. It's a polysaccharide, so potentially it's a good carbohydrate and stop. However, when some structures are recognized, and in this case the hypothesis is that some of those polysaccharides remind plants the polysaccharides of, for instance, of the in the outer shell of fungi. So plants feel that they are under attack, and as a response, they activate some metabolism that otherwise wouldn't be active. And this activation also finally responds in accumulation of stress-related molecules. So in this way, seaweeds prepare plants to respond to abiotic stress through a mechanism that is the same of biotic stress. But again, as I mentioned before, in this case we are focusing on non-biotic processes. This is an example about how this comes from our experiments. I like this example. It's a very simple experiment, but with seaweed extract. But this is the concept. I think that what is the most relevant point we can see here. If you check, this is the yield. Sorry, this was in Italian. I took from a previous slide of mine. If you check the yield, you see when was in hydroponic, when the amount of nutrients is optimal, you don't see differences. You see, okay, you have a slight increase, it's non-significant. On the other hand, it's like, it's a stupid example, but explains well. If you take aspirin and you don't have a headache, you say, okay, I don't need aspirin. But you have a headache and you take the aspirin and say, okay, now I feel better. This is the concept of biostimulants. So you shouldn't use on top of everything. But the concept should be sustainability. So shall I better manage the resources I have? Shall I better manage the possibility to pollute the environment? I can in a way that let me manage inputs. Managing inputs, here you can see, when you decrease to 10% of the nutrients, then you can see a strong difference. The biostimulant shouldn't be like I add a lot of fertilizers and then everything is available, the plant is healthy, no pathogens there. Why using biostimulants? It doesn't make sense. It's just an expense for the farmer. However, when you are under limiting conditions, you see dry biomass, okay, increases also in 100%, but is very relevant when nutrient amount is decreasing. There's another nice example of that one. We had a regional project for using biostimulants on tomato, to implement a strategy for biostimulants in tomato. And we had 3 sites here in this region, in the Emilia-Romagna region. One is pretty close from here, was a farmer that is perfect. I mean, I've never seen, you see, when you see all the plants at the same stage, totally green, any top of the leaves green, so no yellow color, so no chlorosis, everything was perfect because this farmer adds organic matter in the winter and then uses drip irrigation with a lot of fertilizer, so the plants are perfect, but probably this person is overloading. So he guarantees his yield, but he's overloading what are the actual need in the field. And then we had 2 other sites close to the sea here in Emilia-Romagna, in Ferrara, where we have a sandy soil. We have a farmer that makes 3 or 4 cycles per year of different vegetables, so the soil is very stressed. The soil itself is poorer because it's a very sandy soil, it's a bit saline. And the farmer stresses out because just want to harvest 2, 3, 4 times per year. So profitability in short term is okay, but in long term it's not guaranteed, of course, but that person was taking care. Well, the same products, exactly the same products, on the same varieties of tomato, we had 3 varieties in one place, 3 varieties in the other one. In the second site, biostimulants were working perfectly. We could see, perfectly is a big word, but we could see very good results, so an improvement in yield, all the photosynthesis was much better, so is what we expected. In the first site, no responses. So this is the concept, the idea that is also here about biostimulants. Now, at that time, nitrogen was really costly for farmers because urea production was very, very high. The price of urea is linked to the price of gas. So 2, 3 years ago, it was a problem. Now it's not a problem. Maybe it will be a problem again in the next future. But in general, when we speak about sustainability, the paradigm of increasing nitrogen is not anymore working. And in this sense, biostimulants are interesting. A third class of biostimulants, I'm just introducing you, are protein hydrolysates. Protein hydrolysates, again, it's a broad class. So each company has its solution with advantages, disadvantages, but we are not providing nitrogen. So you're not applying 50 kilograms per hectare, whatever it is. We are applying 1 kilogram product. We have bioactive molecules. Protein hydrolysates are taken by cheap sources, can be legume plants or by products from the food industry of animal or plant origin. Anything in principle that is convenient, sustainable from an economical point of view. It's also a matter of circular economy, so can have also some advantages in terms of environment, but for sure the aim of the producers is to have a cheap source to keep the price of the product, the final price of the product low enough to be competitive on the market. And which are the characteristics of protein hydrolysase? Hydrolysis can be made by chemical hydrolysis or by enzymatic hydrolysis. And these two, starting from these two, are completely different. Protein hydrolysase, from chemical hydrolysis, they are often hydrolyzed to amino acids. While when the hydrolysis is enzymatic, we also have peptides, and peptides sometimes are bioactive. This is one big difference, the first one. So the amount of free amino acids and peptide, the ratio, or if you prefer, the degree of hydrolysis. The second point is that you know that amino acids are chiral molecules, are L amino acids in nature. And by chemical hydrolysis, you have racemization, so part of the L are converted into the D amino acids. But in nature, only the L are active. So this is the second big difference. When there's protein hydrolysase, this is the only limitation we have it's in Italy, but it's being adopted across all Europe. When biostimulants, protein hydrolysase, come from byproducts of animal origin, then we have some limitations. We have a specific regulation that states that protein hydrolysate of animal origin cannot be used on edible parts. So you can use, for instance, by drip irrigation, but not by spraying, and cannot be used in organic farming. These are the two limitations, are political decisions, but still, that's the case nowadays. How do protein hydrolysates work? They have, the processes are similar to the previous one, so they increase nutrient use efficiency, so promote growth, promote tolerance to abiotic stresses, and the quality of the products. Just some examples. This one comes like the previous one. When you apply 2.5 ml per liter is a protein hydrolysate that is 0.25%. So it's a small amount. When applied at this rate, chlorophyll, SPAD correlates with chlorophyll. Chlorophyll accumulation is higher. Nitrogen uptake was significantly higher, so the plant performance was better, was improved compared to the same condition where the biostimulant, the protein hydrolysates in this case. Similarly, protein hydrolysates have been known to modulate in a positive way the uptake. You know that this is the GS-GOGAT system in biochemistry, is the way according to which plants incorporate inorganic nitrogen into organic nitrogen, that then is amino acids in the first row, and then amino acids supply with organic nitrogen to other components of the plant. This is in very, very few words how it works. Protein hydrolysates modulate this nitrate reductase activity, that is the first step to convert nitrate into ammonia, and then ammonia is incorporated into organic molecules by the GS-GOGAT. So in this way, the hydrolysate can improve nitrogen uptake that correlates with nitrogen use efficiency. In this other experiment, we have been trying to modulate the resilience to salinity. Again, this comes from an experiment. This was the very first experiment I did with the biostimulants. Just a few words to explain this story. At the beginning, when I started so many years ago working on biostimulants, I was a bit skeptic because actually some products provided nice evidence, some other was totally not working. So there was a debate, okay, it's just a story that will end in a few years, or in future they can be used in a much better way. So everybody has its own opinion at that time. I did this trial. You check control under salinity and control with the hydrolysate. Even visually you can see there are differences, but here when you check the yield, so how the plant, this was lettuce, how the plant could grow under salinity conditions, under growing salinity conditions. This is control and salinity. So salinity decreased 1/3 the yield, more or less. When you apply the product at root level, the decrease was half. When you applied both at roots and leaves, the decrease was 1/3. So it's not completely mitigating salinity, of course, and this is a mild salinity. 25 millimolar is a realistic value you can have in a very saline soils. It's not an extreme condition. Still, it's a challenging condition. So the difference between this and this is made by biostimulants. Salinity determines, as a first row, an accumulation of oxidative stress molecules. And in this way, in this sense, the protein hydrolysates were able to induce the accumulation of phenolic compounds and carotenoids that are two classes of molecules that are well known to be involved in the response to oxidative stress. But this is not an example of mine, it comes from a group in Japan, but it's very nice, and it's the concept that I think should be a biostimulant. Normally, when you have biostimulants, you say, okay, the raw material I use is this one, but you don't have an active ingredient like you can have in pesticides. It's probably a class of molecule that work. In this case, a smart group in Japan could identify a specific peptide in protein hydrolysates that works as a biostimulant. This was called RHPP, root hair promoting peptide. It's a signaling peptide. It's the concept of signaling I mentioned before, because when, very clear here, or if you check this one compared to this one, B versus A, when the biostimulant, this specific peptide, was applied to roots, activate some metabolic processes, modified phytohormones profiles in a way that root hairs were induced. And root hairs are, of course, positive. This is the sequence. So you see, it's a very small peptide. It's 10 or something like that amino acids. This is the sequence. And this one has been then patented and became a biostimulant. This is the exception. In the very most of the cases, we know they work. We know that by optimizing the product, using maximizing this fraction or this other fraction, you can modulate the activity. But normally, it's pretty difficult to link a specific profile or one molecule to a specific activity in biostimulants. Hydrolysates, protein hydrolysates, were also good as peptides in complexing elements and modulating plant nutrition. So they can form some complexes with some minerals. If you think about the side chain of amino acids, you know that there can be positive charges, negative charges. So different specific peptides have the ability to modulate in different way different cationic or anionic species, therefore modulating the availability, therefore modulating the ability to uptake, the bioavailability. There are just examples that run pretty fast because, of course, you could see so many. If you just search in literature, you find thousands of examples. I'm just taking some of them from some other activities. Also, in terms of tolerance to abiotic stresses like high temperature, drought, salinity, and low and high, now also high light conditions, in all the cases, you can find good examples in which different biostimulants can be used. The important point here, this is an example, but what I would like to stress out is that it's wrong to say that a biostimulant works for everything. And now the market, fortunately, is moving to addressed solutions. So this biostimulant works maybe for drought. You cannot simply say, I apply the biostimulant, I don't have to irrigate, I don't have to provide fertilizers. This is a wrong story that somebody uses. The true story is like in pesticides. You know that this fungicide has this spectrum. So it's active against Plasmopara and not against another fungal pathogen. And this same concept should be applied to biostimulants. We cannot simply think that, okay, I spread this protein hydrolysate, I come back home and I don't care about my field and the yield is guaranteed and so on. Are a complementary strategy. And one project we recently presented is about the considering biostimulants in an IPM enlarged view. When you have an integrated pest management, you can integrate also with biostimulants. So having a designed approach for your crop, in your area, in your condition, the right biostimulants against the proper stresses. Interestingly, and this is among the few, protein hydrolysates also increase the quality. Since the functional quality we perceive comes together with the response to abiotic stress, when you increase the resistance to abiotic stress, often you can also increase the quality in terms of sugar accumulation, in terms of antioxidants, in terms of firmness, because the fruits better resist to pectin methylesterase, so the softness that fruits in post-harvest feel as over-ripening, so shelf life is increased. All these aspects are quality aspects for which the farmers are willing to receive some money. This is just an example. I conclude with the last one, mycorrhiza. Mycorrhiza, we could speak about mycorrhiza for days, and it's the oldest story in terms of biostimulants. This is a very well-established symbiosis at root level between these fungi, these mycorrhizal fungi, and plant root. And this is normally happening in nature. It's not something we discovered. The very most of roots are mycorrhized, excepting for few species, mainly Brassica and Chenopodiaceae plants. If we exclude these and very few others, all the other plants in nature are naturally in symbiosis with mycorrhiza. And this symbiosis is specific. So we have this association according to which this strain of mycorrhiza can interact with this type of plant, or crop in our case. And this interaction is mutually beneficial. It has been discovered a few years ago, has been published in a very nice journal, how plants take photosynthate to produce fatty acids. Fatty acids are released by root exudation to mycorrhiza to feed them with carbon. For plants, it's easy to get carbon. They do photosynthesis, of course. It's not costless, but it's convenient. At the same time, mycorrhiza mobilize nutrients, phosphorus in primis, iron then, and in part also nitrogen, and provide those elements to plant. It's a classical symbiosis. And this symbiosis works very well. This symbiosis provides with a large amount of advantages. This symbiosis takes place also naturally because plants release chemoattractants, molecules released by roots that attract mycorrhiza. And in this initial interaction, the two then realize that they have a mutualistic effect, so that one can take benefit from the other and vice versa. And this is the starting of the symbiosis. Starting from that, this symbiosis is so strong, and that's why I call it arbuscular mycorrhiza. This symbiosis is so strong that the hypha can even enter the cells at the root level. So are not simply in the rhizosphere, like several other microbes. That is still important, but this is even beyond. It's a symbiosis in which the hypha, I might have some pictures later, no I don't have, but I can explain in this way. The hypha enter the apoplast, so it's really a tight symbiosis. They exchange molecules. One provides inorganic nutrients, the other one provides carbon, and at the end of the day, both take advantage of the symbiosis. If you check the root architecture compared to without mycorrhiza, the architecture is completely different. In general, mycorrhized roots, they have a much stronger development, and this is one of the reasons for which mycorrhiza increase the water use efficiency, because the plant can explore a larger portion of soil, and therefore takes the benefit to absorb water in a more efficient way. But also increases the amount of fine roots, the fraction of smaller diameter roots that are the most bioactive. So it's not only more developed, is also more active. And again, this promotes water and nutrient uptake, especially those nutrients that are not so available. It is now well recognized that mycorrhiza have a very good phosphatase activity. So phosphate is present in an insoluble form in soil, and therefore it's not available. It's there. The amount is pretty large in several soils, the very most of soils. The amount is significant, but this amount is not available. And if it's not available, plants cannot access it. Mycorrhiza have a good phosphatase activity that release inorganic phosphorus and make phosphorus available to plants. And that's all. Any question?
I have a question.
Yeah. I was asked you to use this one because in the recording then it's available, if you don't mind.
So talking about like grapevine specifically, that stress can be like really beneficial and sometimes desired for having a better quality like in the final grape wine. How can you assess where like when biostimulants are an option? And like I know they probably help with the response to this abiotic stress and can be beneficial on the long term, but how do you assess this decision?
Grape wine is a peculiar crop compared to the others, of course, because there you don't need to maximize so much yield. You need a specific quality, and it's a very good question, your question. There are examples in which some biostimulants, since they modulate the phytohormone profile, they also they delayed the ripening. That nowadays is very important because one of the problems was that sugars were too high and acids too low, even by anticipating the harvest. So if you can somehow modulate, this is a way in which you modulate quality, apart in other crops would be negative. In grape, in this specific context, becomes positive. And this is one of the examples. Another example is that in some area where grape, where now, because of climate change, start to experience water deficit. And there are some, even here in northern Italy, in the Veneto region, where they produce Prosecco. I was speaking with one of the managers that manages the fields there, and he was saying, we never irrigated water. Now we have in some specific fields, we have water stress in grapes. It was something unknown until a few years ago. This could be another example where managing this type of stress can have beneficial effects. Welcome. Some more? Don't be shy.
So one is compared to the use of microorganisms, what you can say about? I mean, we expect the same effect or analogous effect with the chemicals that you show. That's the first question. Second question is concerning. I mean, based on what you said, I mean, it's clear that with this kind of technology, we really need kind of a precision agriculture, precision farming. How could we reach such high level of professionality for doing this? For of course for obtaining then the output that we wish. And the third question is concerning, maybe I miss it. So because it's so specific, so precise the application, do we wish to have a priority in foliar application compared to the soil application, or that is up to the chemical or to the microorganism that we are using as biostimulant? Thank you.
Grazie, Ettore. Are very relevant questions. The first one is about microbials. The examples I provide are non-microbial, of course, because I work much more on that, but we had several experiences also in microbials. With microbials, there are two main limitations now. The first one is the legal aspect. So up to now, only the 4 that were in the regulation can be used. So some companies have maybe good candidates, but it's difficult to develop them. And waiting for the regulation to change, that might take some time, bureaucracy in Europe takes time. In the meanwhile, several of them are moving from microbes to postbiotics. So is the fermentation broth of the microorganism. Because postbiotics, that means the medium in which the microbes are grown, then the microbes are killed, are simply hydrolyzed or filtered, and what remains is used as biostimulant. Because in some cases, these microbes directly work, but in several other cases, the microbe produces some molecules that then stimulate the plant. So when
It will be very similar to the hydrolyzed proteins or not?
Yes, as a concept, yes, this can easily enter the market. Not the microbe, but the postbiotic can enter the market more easily. Because this comes with a second problem with biostimulants, that is formulating them. The farmer, practically, you cannot ask the farmer to keep the product in fridge. It's non-practical, they have maybe large extensions. And formulating biostimulants, maintaining viability over time under like the room temperature conditions, maybe the farmer keep the products under close to the field, so in summer at 30, 35 degrees or even higher in some regions. And this decreases viability of microbes. So if you enter the market, you have to guarantee that they are stable enough. This is the second problem that has limited the spread of microorganisms. Again, for this reason, the tendency is to move in the direction of postbiotics. In some cases, they are even they don't need oxygen. They grow without oxygen. So formulating and keeping these conditions just before application is not something that the farmers are ready to manage. They are in a hurry, they are maybe not expert, and I come to your second question. It's true, it's a precision agriculture now, and they work well when they are used in the proper way. The main limitation is what several companies are investing now is in knowledge. So let the farmers know how to use them. Not simply, I deliver this one, I've done my job, bye-bye. But several companies like Syngenta, that is growing fast in biostimulants, they have technical people that go to the farmers and explain why and how you should apply this product. And this increases the final effects, of course. It's a long trip because some farmers are probably more open, some others are less open, and in past, some of them that tried products that were in the market with no regulations at the very beginning, then they become very skeptic. They say, okay, I tried, I don't want to try anymore because it doesn't work. It might work, but you have to use it in the proper way. I spent 5 years in a private company producing pesticides. At that time, this is a story that fits well with this one. We had a very nice biopesticide. It was a virus for a pest. So it's a very good way of controlling this insect pest. So it was pretty sustainable. When our experimental people in the field tried the product, it was working not like a chemical pesticide, but pretty well. So we were satisfied. You know what was the problem is that you have the window to apply the product was like a few days when the insect reaches a given stage. And when our farmers tried in an experimental station, they could use it to monitor insects and so on. So they applied in the right window and it was working perfectly. But this is not what the farmer in practice does, or at least not so many still do that. And therefore, they were applying like a calendar as they used it to do with classical pesticides. It was not working. So this product exited the market in 2-3 years. I hope this will not be the story of biostimulants, of course. And right for that, it's clear that investing in knowledge for farmers, so for instance, they have to know that when they find this CE, beyond there is a dossier that has been evaluated by you, and it's something that maybe not all of them know. So try to privilege CE products. Try to use them in the right way. They are not the solution for everything, but they can provide some advantages, are part of a world strategy for sure. And sorry, the third question was about, why foliar or, yeah. Again, it's like, I come from the world of pesticides, so I like to make examples from that world, but in pesticides, you have, where to apply, the dosage, and how to, and they're formulated in the proper way. Now, the most serious companies in this field test different dosages, different applications ways, and suggest advice for the best way. Not all of them are good for foliar application. Not all of them are good for drip irrigation. Drip irrigation is convenient because when the farmer irrigates, they just add the product. So they try to privilege for a technical reason, for a practical reason, but some of them still work as spray products. And in this way, it's important. Another example, I met a farmer that was using trichoderma and used it to do the tank mix with fungicide. So if you apply a fungal species with a fungicide, of course you're applying that. But of course it's not his fault. He just has to be trained that he shouldn't, if you apply foliarly, you should apply in a different window from fungicides, otherwise it doesn't make sense. Mycorrhiza, again, some people use the seed-treated seeds with fungicides and then added mycorrhiza. But mycorrhiza is a fungus, so they don't match one with the other, of course. Those are part of a long trip, I think. The biostimulants at the beginning, pesticide, you know the story of pesticides better than me because you lived this story. At the beginning, the regulations were a bit wider than strict, and strict, they are even extremely strict. Biostimulants are taking the same story. So at the beginning, entering the market was just prepare a statement, okay, I use this one, it's natural, okay, let's go on. That shouldn't be the way. Now they are increasing the level, both from the side of the producers and from the side of the farmers, because both need to do their part. And when both work in the same direction, then the final outcome is positive.
Okay. So thanks a lot, Luigi.
My pleasure.
Yeah. And good luck with the students.
I will forward you the presentation you can share with the students.
What the tea leaves say, the Roundtable Research. An open tea podcast, communication and dissemination services.